let's continue our exploration of bioenergetics by delving into how glycolysis leads to the Krebs cycle. This pathway is crucial for understanding how our bodies produce energy during exercise, especially when oxygen is readily available. When oxygen is present in sufficient quantities within the mitochondria, the specialized cellular organelles where aerobic metabolism occurs, the end product of glycolysis, pyruvate, is not converted into lactate. Instead, pyruvate is transported into the mitochondria for further processing. Along with pyruvate, two molecules of reduced nicotinamide adenine dinucleotide, or NADH, produced during glycolytic reactions are also transported. The term reduced refers to the addition of hydrogen atoms to NAD+, forming NADH. Once pyruvate enters the mitochondria, it is converted into acetyl coenzyme A, commonly known as acetyl-CoA. This conversion is facilitated by the pyruvate dehydrogenase complex and results in the loss of one carbon atom from pyruvate as carbon dioxide. Acetyl-CoA can then enter the Krebs cycle for further ATP resynthesis. Meanwhile, the NADH molecules enter the electron transport system, where they can also be used to resynthesize ATP. The net reaction for glycolysis when pyruvate is shuttled to the mitochondria can be summarized as follows. Glucose plus 2 inorganic phosphate molecules plus 2 molecules of adenosine diphosphate plus 2 molecules of NAD plus yield 2 molecules of pyruvate, 2 molecules of ATP, 2 molecules of NADH, and 2 molecules of water. So now, let's discuss the energy yield of glycolysis. There are two primary mechanisms for resynthesizing ATP during metabolism: substrate level phosphorylation and oxidative phosphorylation. Phosphorylation is the process of adding an inorganic phosphate to another molecule. For example, when adenosine diphosphate combines with an inorganic phosphate, it forms adenosine triphosphate. 2. Oxidative phosphorylation refers to the resynthesis of ATP in the electron transport chain. In contrast, substrate level phosphorylation refers to the direct resynthesis of ATP from ADP during a single reaction within the metabolic pathways. In glycolysis, there are two steps that result in substrate level phosphorylation of ADP to ATP, the conversion of 1,3-bisphosphoglycerate to 3-phosphoglycerate, catalyzed by the enzyme phosphoglycerate kinase. In this reaction, one molecule of ADP is converted to ATP, the conversion of phosphoenolpyruvate to pyruvate, catalyzed by the enzyme pyruvate kinase. Here, another molecule of ADP is converted to ATP. The gross number of ATP molecules resynthesized as a result of substrate level phosphorylation during glycolysis is 4. However, it's important to consider that ATP is also consumed during glycolysis. The reaction that converts fructose 6-phosphate to fructose 1,6-bisphosphate, catalyzed by the enzyme phosphofructokinase, so PFK, requires the hydrolysis of one ATP molecule. Additionally, there are two possible sources of glucose. Blood glucose. When glucose enters the muscle cell from the bloodstream, it must be phosphorylated to remain in the cell and maintain the glucose concentration gradient. This phosphorylation is catalyzed by the enzyme hexokinase and requires one ATP molecule. Muscle glycogen. When muscle glycogen is broken down into glucose, a process called glycogenolysis, with the help of the enzyme glycogen phosphorylase, the glucose is already phosphorylated. Therefore, it does not require the hydrolysis of ATP. As a result, starting with blood glucose, 2 ATP molecules are used, one by hexokinase and one by phosphofructokinase. 4 ATP molecules are produced through substrate level phosphorylation. This results in a net gain of 2 ATP molecules. Starting with muscle glycogen, 1 ATP molecule is used by phosphofructokinase. 4 ATP molecules are produced. This yields a net gain of 3 ATP molecules. Understanding these differences is crucial for appreciating how energy yield can vary depending on the source of glucose. Now let's explore the control of glycolysis. The rate of glycolysis is generally stimulated to increase during intense muscle actions by high concentrations of ADP, high concentrations of inorganic phosphate, increased levels of ammonia, slight decrease in pH, elevated levels of adenosine monophosphate. These factors are signs of increased ATP hydrolysis and the need for more energy. Conversely, glycolysis is inhibited by significantly lower pH, more acidic conditions, high concentrations of ATP, high levels of creatine phosphate, elevated citrate levels, increased free fatty acids. These conditions are usually present at rest. It's important to note that while a slight decrease in pH can stimulate glycolysis, a significant decrease will inhibit it. More specifically, the regulation of glycolysis involves the concentrations and turnover rates of 3 important glycolytic enzymes: hexokinase, phosphofructokinase, and pyruvate kinase. These enzymes are regulatory because they have important allosteric binding sites. Allosteric regulation occurs when the end product of a reaction or series of reactions binds to an enzyme at a site other than the active site, influencing the enzyme's activity. This process is also known as end product regulation or feedback regulation. Allosteric inhibition happens when an end product binds to the regulatory enzyme and decreases its turnover rate, slowing product formation. Allosteric activation occurs when an activator binds with the enzyme and increases its turnover rate. For example, hexokinase, which catalyzes the phosphorylation of glucose to glucose 6-phosphate. It's allosterically inhibited by high concentrations of glucose 6-phosphate in the sarcoplasm. This means that when glucose 6-phosphate levels are high, hexokinase activity decreases, preventing an overaccumulation of glucose within the cell. Phosphofructokinase PFK is the most important regulator of glycolysis because it controls the rate limiting step. ATP acts as an allosteric inhibitor of PFK. As ATP levels rise within the cell, PFK activity decreases, slowing down glycolysis. Conversely, AMP acts as an allosteric activator of PFK, stimulating glycolysis when energy is needed. Pyruvate kinase, the enzyme that catalyzes the final step of glycolysis, is allosterically inhibited by ATP and acetyl-CoA, an intermediate of the Krebs cycle. It is activated by high concentrations of AMP and fructose 1,6-bisphosphate. Understanding these regulatory mechanisms helps explain how glycolysis adapts to the energy demands of the cell during different intensities of exercise. Now, let's address a common misconception regarding lactic acid and muscle fatigue. It's often believed that lactic acid causes the burning sensations experienced during high-intensity exercise, a condition termed lactic acidosis. However, this is a misnomer. The production of lactate during glycolysis does not directly contribute to acidosis. In fact, the conversion of pyruvate to lactate consumes protons, H+ ions, which can actually help buffer the cell against acidity. The main source of proton accumulation leading to metabolic acidosis during intense exercise is the hydrolysis of ATP outside the mitochondria, not the production of lactate. Therefore, the term lactic acidosis is inaccurate. The appropriate term is metabolic acidosis, which describes the reduced pH within skeletal muscle during high intensity fatiguing exercise. Returning to our discussion on energy systems, let's explore the lactate threshold and the onset of blood lactate accumulation, or BLA. As exercise intensity increases, there are specific breakpoints in the lactate accumulation curve. The lactate threshold is the exercise intensity at which blood lactate begins an abrupt increase above baseline levels. It represents a significant shift toward anaerobic metabolism to meet energy demands. In untrained individuals, the lactate threshold typically occurs at 50% to 60% of maximal oxygen uptake. In aerobically trained athletes, it occurs at 70% to 80% of maximal oxygen uptake. A second increase in lactate accumulation occurs at higher intensities, and it's termed onset of blood lactate accumulation, OBLA, defined by blood lactate concentrations reaching 4 millimoles per liter. Training at intensities near or above the lactate threshold or onset of blood lactate accumulation can shift these thresholds to the right, meaning lactate accumulation occurs at higher exercise intensities. This shift is likely due to changes in hormone release and increased mitochondrial content, enhancing the muscle's ability to produce ATP aerobically. Now let's explore the oxidative aerobic system, the primary source of ATP at rest and during low intensity activities. This system predominantly uses carbohydrates and fats as substrates. Protein contributes minimally to total energy production, except during long-term starvation or prolonged exercise exceeding 90 minutes. At rest, approximately 70% of ATP production comes from fats and 30% comes from carbohydrates. As exercise intensity increases, there's a shift toward greater carbohydrate utilization. During high-intensity aerobic exercise, almost 100% of energy is derived from carbohydrates if sufficient supplies are available. Glucose and glycogen oxidation begin with glycolysis. If oxygen is sufficient, pyruvate from glycolysis is transported into the mitochondria and converted to acetyl-CoA by the pyruvate dehydrogenase complex. Acetyl-CoA enters the Krebs cycle, a series of reactions that further oxidize substrates to produce ATP indirectly via substrate-level phosphorylation. For each molecule of glucose, the Krebs cycle produces 2 ATP molecules indirectly from guanine triphosphate GTP, 6 molecules of NADH, 2 molecules of flavin adenine dinucleotide, FADH2. The NADH and FADH2 molecules transport hydrogen atoms to the electron transport chain, ETC, where they contribute to ATP production through oxidative phosphorylation. In the ETC, hydrogen atoms are passed through a series of electron carriers known as cytochromes, creating a proton gradient that drives ATP synthesis. Oxygen serves as the final electron acceptor, forming water. One molecule of NADH can produce approximately 3 ATP molecules, while one molecule of FADH2 can produce about 2 ATP molecules. Altogether, the oxidation of one molecule of glucose via the oxidative system yields approximately 38 ATP molecules. If starting from muscle glycogen, the net ATP yield is 39 due to the savings of 1 ATP in the initial phosphorylation step. Fat oxidation also plays a significant role in energy production, especially during prolonged low-intensity exercise. Triglycerides stored in fat cells are broken down by hormone-sensitive lipase into free fatty acids and glycerol. Free fatty acids enter the mitochondria and undergo beta oxidation, a process that converts them into acetyl-CoA and hydrogen atoms. The acetyl-CoA enters the Krebs cycle, while the hydrogen atoms are carried by NADH and FADH2 to the ETC. The oxidation of fatty acids yields a substantial amount of ATP. For example, the breakdown of one triglyceride molecule containing 316 carbon fatty acids can produce over 300 ATP molecules. Protein oxidation is generally a minor contributor to energy production. Proteins are broken down into amino acids, which can be converted into glucose, pyruvate, or Krebs cycle intermediates. The oxidation of amino acids is minimal during short-term exercise, but can contribute up to 18% of energy requirements during prolonged activity. The primary amino acids oxidized in skeletal muscle are the branched chain amino acids, leucine, isoleucine, and valine. The nitrogenous waste products from amino acid degradation are eliminated as urea and ammonia. Accumulation of ammonia is associated with fatigue due to its toxic effects. Controlling the oxidative system involves regulating the rate-limiting steps in the metabolic pathways. In the Krebs cycle, the enzyme isocitrate dehydrogenase catalyzes the conversion of isocitrate to alpha-ketoglutarate and is stimulated by ADP and inhibited by ATP. So the availability of NAD+ and FAD2+ is also crucial as they accept hydrogen atoms during oxidation reactions. A shortage of these carriers slows the Krebs cycle. Additionally, accumulation of certain intermediates like succinyl-CoA can inhibit earlier steps in the cycle. The electron transport chain is inhibited by high levels of ATP and stimulated by high levels of ADP, ensuring that ATP production aligns with the cell's energy demands. In terms of energy production and capacity, the phosphagen system provides the fastest rate of ATP production, but has the lowest capacity due to limited stored ATP and creatine phosphate. The oxidative system has the slowest rate but the highest capacity for ATP production, making it essential for endurance activities. There's an inverse relationship between the rate of ATP production and the capacity of each energy system. The phosphagen system supplies ATP for high-intensity, short-duration activities like a 100-meter sprint. The glycolytic system supports moderate to high-intensity activities of short to medium duration, such as a 400-meter run. The oxidative system is the primary source of ATP during low-intensity, long-duration activities like marathon running. It's important to recognize that all three energy systems are active at any given time. However, their relative contributions depend on the intensity and duration of the activity. During exercise, energy production transitions smoothly among these systems to meet the body's demands. Understanding how glycolysis leads to the Krebs cycle and how these energy systems interact provides valuable insights into optimizing training and performance. By tailoring exercise programs to target specific energy pathways, athletes can improve efficiency, delay fatigue, and enhance overall performance.